Okay, I mentioned this effective length, right? There's the length of the transcript and then the effective length, and that effective length was always shorter. So I want to just spend one slide telling you what that is, because otherwise I know it leads to questions. So it's easier if I just head this off now. So the effective length is the length of a transcript after adjusting for the total number of possible positions that a fragment of some size x, could originate from. That's kind of complicated to think about, so let's do an example here, and let's actually start with a formula. The effective length is actually calculated by taking the actual length of the transcript, subtracting the length of your fragment that you're trying to figure out if it could have come from that, and you're adding 1 to it. So I like to give examples, so we're going to kind of unpack this a bit. So if we took a transcript that had an actual length of 12 nucleotides, and here's the actual transcript. And again, we're making these things really small and simple so that we can understand them. And let's say we took a fragment of length 3, and we don't care. This isn't an alignment per se. We just want to know how many possible places could a fragment of 3 nucleotides could have come from this transcript of length 12. So our actual length is 12. That's this. Our fragment length is 3. So we already know 2 of these numbers, and we're just going to add 1. So our effective length is really easy to calculate. It's 12 minus 3 plus 1. So the effective length of this transcript, even though the transcript is 12 nucleotides, its effective length is 10 nucleotides. How is that possible? Well, we just have to think about a 12 nucleotide transcript and how many places a 3 nucleotide sort of fragment could come from. Well, it could come from here, ATG. It could come from TGC. And you just keep shifting that line over until you hit the end. And if you count those, it's 10. Now, rather than do that for all these different length transcripts and fragment sizes, there's an actual formula, a very simple one, that gets you to that same number. All right, so the effective length is an adjustment of the actual length, adjusting for the total number of places that a fragment of size x could have originated from. Rather than calculate that for each fragment in our library, we take a distribution of fragment sizes. You remember we actually entered fragment sizes into the Callisto aligner. It uses those metrics we entered when we did the alignment to actually come up with an estimated or approximated effective length. And so if you look back at your abundance.tsv file and you take the length and you subtract the effective length, you'll get a number that is about the size of that number that we put into the Callisto aligner. So hopefully that clears up a little bit this idea of length and effective length. So from this point forward, when I talk about how we sort of express units of gene expression, when I talk about length, I really mean effective length. All right. A really key thing, if you take nothing else away from this lecture, the key thing to understand is that RNA-seq is not quantitative. It's not absolutely quantitative. We never know exactly how many transcripts arise from a particular locus. All we know is relative expression. So RNA-seq is a relative measure of gene expression, and that is essential to understand. There is no exception to that rule that I'm aware of. When you run RNA-seq, it is a relative measure of expression. So I want to kind of pin this concept then for the rest of the presentation, because I think the way we need to think about, talking about relative expression, we sort of need to understand that all commonly used techniques such as, RNA-seq to measure mRNA abundance, qPCR, microarray, whatever, they really should be aiming at estimating a statistic that is as closely proportional to the relative molar concentration as possible. And so that's how we're going to assess which of these kinds of units of expressing count data for RNA-seq is really the best. It's going to be the best one, the one that does the best job of estimating the relative sort of molar concentration, if you will, for all the transcripts in the transcriptome. So I want to emphasize that understanding the units that we use to talk about measuring counts in RNA-seq data is really critical to understanding how we determine differential expression and how we think about how we normalize and what normalization methods are effective. Okay, that then leads us to this very loaded term, very broad term, sometimes misused term of normalization. What does it mean to normalize data? I'm going to tell you how I define it. I don't know that there is a universally accepted definition, but I think of it as a generic term. That's the easiest way I can sort of work with it. It's a generic term that refers to any number of ways that a data set is globally altered. And I feel like that's a key part of this, globally altered in order to improve our ability to detect differentially expressed genes. So with that in mind, maybe there's a number of things that count as normalization, but I'll give you I'll give you some specific examples and classes of ways of thinking of different types of normalization. For those of you who are, savvy on a flow cytometer, for the immunologists who are taking this class, to kind of put things into context, I think of RNA-seq normalization as being kind of roughly equivalent to compensation on a flow cytometer, right? You're globally altering, the way your data is represented in order to better discriminate, single positive, double positive populations on a flow cytometer. Okay, so let's start with an example, and I'm going to use this kind of a format throughout the rest of this first half of the presentation to kind of walk you through with some very simple numbers and some very sort of trimmed down data sets here, what happens to our data when we tweak it in different ways. So in this case, I've got 4 genes down the left-hand side, and I've got 5 samples, 5 mice. And I'm showing that gene A has, very low gene expression relative to these other 3 genes. Gene D has very high gene expression. And I've calculated the standard deviation of that gene expression. And one thing you may notice looking at this is that the gene that's the most lowly expressed has the lowest standard deviation, while that gene D that's really highly expressed relative to the others has the highest standard deviation. This is actually a problem or a challenge, which is that as our gene expression goes up, the standard deviation goes up. And this is actually a plot of a real RNA-seq data set and what this looks like for that data set. Genes that have a higher median expression, each of these points is a gene, have higher standard deviation. That by definition means that it's going to be harder, to find or to pull these genes out as being differentially expressed between 2 conditions or more because they have really high standard deviation. And that's probably not great because if they're really highly expressed, they might be really important in our system. So genes with a higher expression have a higher standard deviation. This has a term in statistics. It's called heteroscedasticity, where the variance is not even across different levels of your expression value, and so that's a problem. So what we can do is take these raw counts, which is what these are. These are raw numbers of reads mapping to these genes. We can take these raw counts and we can convert them or transform them using a log2 scale. All right, and when we do that, now look what happens. So this is the exact same data I just showed you, but it's been transformed by a log2 conversion, and now we see something very different. First of all, these standard deviations are a lot tighter, but now our most lowly expressed gene has the highest standard deviation, and so we've sort of swung the pendulum the other way. This is that exact same data set after log2 transformation. So looking at the median log2 expression versus standard deviation, very different profile. Now actually the genes that have the lowest expression have the higher standard deviation, which is what I just kind of pointed out here. And so that really highlights, I think, a challenge with normalization, at least as I defined it. When you tweak a data set globally, oftentimes you fix one problem while creating another. And it's just a matter of choosing the problem that you're happier dealing with, or the assumptions or the changes that better fit the kinds of things that you want to do. So for example, in this case, the genes that have, the higher standard deviation are the most lowly expressed. Maybe that's okay because maybe these really lowly expressed genes are much less likely to be involved in the biological process we study. Who cares if a gene goes from being almost not expressed at all to just trivially induced upon some condition. So maybe we're okay having more noise down here than we were having more noise up in our highly expressed genes. So this is really why you see so frequently, whether it was microarray data back in the day or RNA-seq data now, that we often talk about it in a log2 scale. It's because it's just much better for plotting, for thinking about, and for representing that data in that way. Okay, now we need to talk about units and how we really sort of attach numbers to our genes or transcripts, because we don't leave that data as counts. We don't leave it as log2 counts. So let's talk about a couple situations. One is, let's kind of give ourselves some context here. We've got 8 samples that we prepared libraries from. We pool those samples, we sequence those samples, and we generate a single FASTQ file that gets demultiplexed because we have each of these 8 samples. We have indexes or indices on them, and we can call those barcodes too. Remember, that language is really the same. And so using those sample-specific barcodes, we can demultiplex them. Often that happens on the machine, but we can also do it manually. But in the end, we take this single FASTQ file and we have 8 FASTQ files now. Now, in an ideal world, expected number of reads per sample would be even for all 8 of those samples, right? Put 8 samples, put them together, we sequence, we pull them back apart, it's equivalent. But we know that we're not that good at pipetting. We're not that good at measuring really small amounts of DNA. And so what we actually end up with, right, is something more unbalanced, where we have FASTQ files that do not have the exact same number of reads in them. And so that has a real consequence in terms of how we think about counting, assigning reads to genes and measuring the expression of that gene. So here's an example. Here's 2 samples, 2 FASTQ files. One that, I've indicated with green has a bunch of reads, and one that doesn't, has much less, maybe half as many. So let's look at the same gene. I'm going to just move forward here. Let's look at the same gene. We're going to call this gene A, and it's gene A in sample 1, and it's gene A in sample 2. That gene has 3 exons. If we're to look at that gene in sample 1, it will have twice as many reads aligning to it. Not because it's more highly expressed in this sample, in sample 1 compared to sample 2, simply because we sequenced that sample more deeply. The sequencing depth was greater for that sample or for that condition. And so as a result, if we were to just kind of look at the data without changing it in any way, we might erroneously come to the conclusion that sample 2 was, expressed at half the level as sample 1. The gene in sample 2 was expressed at half the level in sample 1. That some condition had an effect on the expression of that gene. So, however, if we account for that sequencing depth, because we know it, right? We know that this sample had, say, 40 million reads and this had 20 million reads. If we divide the sequences, the reads mapping to each transcript or gene by the sequencing depth, then we now sort of account for that, and now we have a much better estimate of the relative expression of this gene between these 2 samples. And by the way, I call that, scaling for units gene expression between libraries, between library normalization, if you will. But that's not enough. We also oftentimes think about scaling units within a sample. Why? Well, if we take just that single sample that had 40 million reads, and we look not at one gene, but at 2 different genes, then if gene A is a 3 exon gene that's quite long and gene B is a small 1 exon gene, just by virtue of the gene length, and I'm going to use gene and transcript interchangeably here because it doesn't matter for this purpose. If those 2 regions are very different in size, then they will have different numbers of reads mapping to them. The smaller region will generate fewer fragments for sequencing. The larger region has more of an opportunity to have reads aligning to it. And so again, if we were not correcting for that, we would interpret gene A as being more highly expressed than gene B, when in reality, if we actually put a fixed window length over there and normalized the read depth to a unit length, then we would control for that. All right. And indeed, that's exactly the basis of what you probably have heard quite a lot about, which is RPKM, reads per kilobase, so scaling by length, per million reads in the library, so scaling per sequencing depth of the sample, RPKM. And that's really effectively the same as FPKM, which FPKM is just used when we're talking about paired-end data. So RPKM or FPKM, same thing. You first adjust or normalize between samples and within a sample. And if you're interested in that original reference, they're here. But for a long time, sort of post 2008, that was really what people meant, I think, when they would talk about normalization. And it was not uncommon to see, papers that published, in fact, it was common to see papers that published just RPKM or FPKM values with no other adjustments. So what we're going to do now is walk through why that's no longer sort of the accepted approach for attaching units to gene expression, and how we can do a better job of expressing these units now moving forward. So RPKM or FPKM is really just a unit of expression for RNA-seq data. We can quibble about whether it's a normalization method. I don't think of it as a normalization method per se, but it is just a way to take those original counts and adjust for the length of the feature and the depth of sequencing of the sample. All right, so that means that RPKM is the number of reads mapped to a genomic region divided by that region's length in KB and the total times the total number of reads. So let's take an example, 1,000 reads mapping to a 5 KB region from a library that had 20 million reads. That would give us an RPKM of 0.00001. So that's really, really small, and biologists tend to really feel uncomfortable about tiny numbers, sort of inherent distrust of tiny numbers. Very, very different from physicists, for example. But so we take that and we multiply it by 1 million, so this arbitrary multiplier, to take this number and turn it into something that now we feel really good about, right? That's 10. That's an RPKM of 10. That sounds way better than 0.00001. But this really is sort of arbitrary. And this multiplier, sometimes you see it here at the end of the formula. Sometimes you see it baked in here. So instead of total number of reads, it's total number of millions of reads, right? But in the end, it's all to keep our numbers in a range that we feel comfortable with. But the issue is that we now know, for a long time, we've known that RPKM alone really is not sufficient for normalization. If you look at RPKM and you ask how it compares to sort of what I'll talk about later of sort of true normalization methods, between sample normalization methods, it doesn't do as good of a job with variance, with housekeeping genes, with clustering, with false positive rates. In almost every way, or at least every way based on this table, it's inferior to methods employed in EdgeR and DE-seq and Lima. Lima is one of the differential expression tools that we'll use. So this is why I say RPKM and FPKM now is sort of not the accepted standard. So why? Why is RPKM so bad? It seems totally logical, right? Scale by unit length and then scale by sample sequencing depth. So we'll again use a toy example to demonstrate sort of where things go awry with RPKM. So here's 2 samples, and I've got samples down the left and genes across the top now, and 5 genes. And I've indicated the length of those genes here. And again, whether we're talking about transcripts or genes doesn't matter. And then here's the number of reads mapping to each of these genes in the 2 samples. And I've tallied up the total number of reads. So this would be like your library This would be your sequencing depth on the sample. 100 reads deep on this sample, 500 reads deep on the second sample. All right, so remembering that formula I showed you, if we take a look at just like keep our eyes on one of these, the 80 reads mapping to gene A in sample 1, that now becomes 8,000 reads mapping to gene A in sample 1, 8,000 RPKM mapping to gene A in sample 1. We can see our total RPKM here, 28,000, versus in sample 2, 822,000. So remember what I told you, RNA-seq is supposed to give us a relative quantification of each gene expression, and it should be as closely proportional as possible to the relative molar concentration. The average relative molar concentration then for each and every sample, if it's mapped to the same reference genome, should be the same, right? If we think of that reference file as our pot, right, our bucket, that reference FASTA file, and we map reads to that bucket, the relative molar concentration, right, the total, total bucket size should be the same. And so if we look at this now, this was that original reads mapping and our RPKM estimates from those. If we figure out the average, if we think of RPKM as hopefully being a good relative molar concentration, our average are very different, right? Our average across these 5 and these 5, very, very different. And so it at least by the measure that we talked about of what a good way to talk about relative gene expression would be, RPKM seems not to be ideal. And so how do we fix RPKM? Well, fixing it's pretty easy. We just kind of change the way we come up with the unit. So I'm going to walk through that now. So we're going to use that same data. Here's that 80. We have 100 total reads, 500. I'm going to scale by gene length first. So I divided 80 by 100 to get 0.8. I divided 10 by 50 to get 0.2, etc. And then I tallied up that row to get a total RPK. This is your reads per kilobase. All right. This total RPK now becomes a normalization factor, and we can divide all of our sample, the reads per K, by that total RPK in the sample. That is the formula for transcripts per million. That's the formula that was used to generate the data you see in the Callisto file. So what's that look like? Well, we take that 0.8, we divide it by 2.84, 0.2 by 2.84. For the second sample, we're dividing by this total RPK for that sample, so 0.2 divided by 411, et cetera, and that gets us some really small numbers again, right? But look at this. They total up to the same. They're total to 1. So the bucket now is exactly the same. For sample 1, if we add up the abundance of all the transcripts or genes measured in that sample, they add up to 1. The pi is 1. That's exactly what we're shooting for, right? Now, we have the same issue with, some inherent distrust of small numbers for biologists. So we add in our multiplier, we kick these numbers up, and now now they total to 100. So transcripts per million. That's exactly what we're talking about here. Very simple formula, a really simple way to get to them. And they meet now this criteria of being, that the relative molar sort of concentration should be, the average relative molar concentration should be the same between two different samples if they're mapped to the same reference file. And there we go. There's the average, right? 1 million divided by 5 is 200,000. Same here. Okay, so that's great, right? We've now got this new way of talking about units for gene expression, TPM. We feel like it's going to be sort of better for talking about relative gene expression than RPKM, but we're not done. So further normalization really is warranted before we can compare between samples. So none of the things I just showed you, scaling to unit length, doing scaling to sequencing depth, doing TPMs, none of those really give you license to compare between samples. And so, well, let's take a look at this. So this was a really seminal paper in 2010, and the way they articulated it, I think, is better than I probably could, so I'm just going to read through this. Library size scaling, so things like RPKM, is too simple for many biological applications. the number of fragments expected to map to a gene, is not only dependent on the expression level and length of the gene, right? But also the composition of the RNA population that's being sampled. The composition of the RNA population that's being sampled, and I'll show you why that is. Thus, if a large number of genes are unique to or highly expressed in one experimental condition, the sequencing, so-called real estate, available for the remaining genes in that sample is decreased. If not adjusted for, this sampling artifact can force the differential gene expression analysis to be skewed towards one experimental condition. This is really critical. So, as a simple way of describing this, if I only have 400 million reads coming off my sequencer, that is a fixed amount of real estate, all right? And if I have one sample that has 40 million reads, that's a fixed amount of real estate. If I go and change dramatically the level of one or more genes, there are, by definition, fewer genes left to go around to the other, fewer reads left to go around to the other genes. So as an example of this, let's look back at those tables I just showed you, where we went from counts for these 5 genes and we calculated reads per kilobase to get this total RPK, and then we used this formula to calculate TPM, and we were really happy, right? Well, I don't know if anybody caught this in the tables, but there's a real problem here, right? So take a look at this gene D. Clearly, if sample 2 was a different condition than sample 1, that condition totally induced, much higher expression of gene D. It went from 3 reads mapping to it to 50, and it didn't change in size, right? So it's much more highly expressed in sample 2. And yet, by the time we get through to our TPMs, it looks like it's dramatically downregulated, tenfold downregulated nonetheless, right? It's more than tenfold upregulated here by raw counts, but more than tenfold downregulated by the time we get to our tried and true unit of expression, TPM. Why, right? Differential expression across this whole table is kind of wacky. Why is it so messed up? I mean, you can see, gene C looks like it was induced, as was gene B. And those both look downregulated as well. Gene A is downregulated for sure, but here it looks very, wildly downregulated. Well, the reason is this culprit. We have this gene here that is just really an outlier, right? It went from 1 to 400 reads mapping to it. It had 400 times more reads mapping to it in this condition that this sample came from than this condition that sample 1 came from. All right, so what we can do then is sort of ignore that sample. And that's really, in essence, without getting too much into the details, sort of modern cutting-edge normalization methods try to account for this. They look at these sort of funky genes. And if you imagine sort of ignoring this gene, right? And then we recalculate all these numbers, ignoring this gene. All right, so just tallying up these 5, but only these 4. We get now equivalent total reads. Our RPK now, instead of being 411, right? We've left out this 400, it's just 11. And now we see that, more true to form here, this gene that looked upregulated by counts is upregulated. These 2 genes, one that's upregulated, one that's downregulated, those look much closer now. This one's actually downregulated. This one that was pretty dramatically downregulated now looks dramatically downregulated, not a sort of crazy downregulated. So this is a way to kind of balance things just roughly. And so most current normalization methods attempt to find a set of genes with kind of minimal variance, which is what I basically did here manually, across the samples that you're going to use. And it uses those genes for normalization. And one of those methods, the trimmed mean of M values, is one that's described here. And it's actually the TMM method is the one that we'll use for normalizing our data in R. Okay, just a word of caution. Statistical tools for normalization and for differential gene expression make certain assumptions about your data, right? That's kind of at the heart of statistical methods and how we choose them. each one has some inherent assumptions. And at least for differential gene expression, some of those key assumptions, or at least two of them, one is that most genes, an assumption is that most genes are not going to be differentially expressed in your data set. And so if you have a condition, a sample, a situation where there is wildly different gene expression between your two conditions, that could really change the kinds of data you might get out of a differential gene expression analysis, because it's assuming most genes are not differentially expressed, and of the ones that are differentially expressed, approximately equivalent numbers are upregulated as there are downregulated genes. So there's roughly a balance in who's going up versus who's going down. So if you have a condition where many genes are differentially expressed only in one direction, those just pose different challenges. I'm not saying you can't use the tools that are out there for normalization and differential expression. Just know that some of those key assumptions might be getting violated in your data, and you just have to kind of be aware of that. That's all I'm saying. Okay, so that wraps up part 1. As I said, in the next part, we're going to actually get started in R.